Welcome everyone. Thank you all for joining the G6PD Operational Research Community of Practice webinar. Today's session is on the validation and operational use of point-of-care G6PD testing in the Brazilian Amazon. And we'll be hearing from two presenters today on data from clinical study in real-life settings that inform test performance and considerations for use. So this is today's agenda. We'll start with a quick introduction about the GORCOP, get right into our two presentations, and then we will have time at the very end for questions. So for those who may be new to the G6PD Operational Research Community of Practice, the GORCOP is a collaboration of researchers, organizations, malaria control programs, and clinicians committed to advancing the introduction and scale up of G6PD diagnostics through operations research in support of safe access to radical cure treatment for P. vivax malaria. The group aims to harness best practices and lessons learned, support information sharing and connections among participants, and provide ongoing updates. We maintain a digital repository of resources that are accessible to all of our participants, as well as host meetings and webinars like this one that you're attending today. We do have a webpage for the community of practice, which can be found at the link you are seeing on the screen now. And you can find additional information there. There is a link to join the GORCOP if you are not already a member. And please feel free to follow up with us if you have any questions. So without further ado, I will pass it over to our moderator, Roberta Ataides, who is the Program Director at Global Health Strategies Brazil, to get us into the presentations and introduce our speakers. Thank you.
Many thanks, Amanda, for the introduction, and hello, everyone. Thank you all for joining us today. I'm very happy to participate today as a moderator, so I just would like to take a few seconds to thank Pat for the invitation and also thank the speakers for having me. So just before we really jump into the presentation, I would like to share a couple of operational details about how this meeting will work, how we have planned the presentations today, so we all can be on the same page. So first of all, I think you have noticed, but this webinar is being recorded. So the recording will be shared with all of you after the meeting. Also, as Amanda mentioned, after the presentations, we will have a Q&A session. So if you have any questions, you can type it into the chat, or you can also use the raise your hand function in Zoom so that we can call your name and then you can open your mic and make a question. And if anyone, since Diego and I are Portuguese speakers, so if there is any Brazilian or anyone that feels more comfortable in making questions in Portuguese, please feel free to do so, and Diego and I can help with the translation. So I think this is it. So I think we can move on to our first presentation for today. So our first speaker is Stefan Zobrist. Stefan is a public health researcher with over 7 years of experience in research and program management focused on the development and introduction of global health technologies. She's currently working as a program officer with PATH's Global Diagnostic Program, where she leads and supports clinical and user research for diagnostic products, including evaluation of diagnostic for performance, usability, and operational feasibility. So thank you, Stephanie, for a presentation. Over to you.
Thank you, Roberta. Can everyone hear me okay?
Yes.
Yes.
Great. So my name is Stephanie Zobrist. As Roberta said, I work with the diagnostics team at PATH. And I'll be presenting today on the topic of a clinical validation study of a point-of-care G6PD test that was conducted in Brazil. And just a quick note that although I'm the one presenting today, I'm doing so on behalf of the study team and our partners at FMT and CEPEM in Manaus and Porto Velho, where the study was implemented. Next slide. Next slide. Great. So in this presentation, I'll discuss the findings of the clinical performance study. And first, I'll begin with a brief overview of the test that was evaluated itself for anyone who may be unfamiliar with it. The standard G6PD test is a semi-quantitative biosensor test intended to aid in the detection of G6PD deficiency. And this test is an enzymatic colorimetric assay developed by SD Biosensor, a company based in South Korea. And the test measures G6PD enzymatic activity and total hemoglobin concentration in fresh capillary and venous human whole blood specimens. The components of the test include a portable battery-operated analyzer that you can see in the top right-hand corner, disposable test strips called test devices, and individual extraction buffer tubes, sample collector tubes called easy tubes, and a lot-specific code chip that's used to calibrate the analyzer. And to conduct the test, 10 microliters of blood is collected using an easy tube, mixed with the extraction buffer, and applied to the test device, which is then inserted into the analyzer. After a period of 2 minutes, the analyzer provides a numeric measurement of G6PD activity normalized by hemoglobin, as well as total hemoglobin in grams per deciliter. And then the G6PD measurement can be used in a semi-quantitative manner to classify individuals as G6PD deficient, intermediate, or normal according to the thresholds provided by the manufacturer. Next slide. So here you can just see those thresholds for deficient and normal for males and deficient, intermediate, and normal for females. As has been discussed in prior webinars, the interpretation of this test is semi-quantitative, and the result output is used with these thresholds to determine the G6PD status of an individual. And these are the thresholds that were used on the test as part of this clinical validation study. Next slide. So this slide just shows an overview of the study itself, the Brazil G6PD point of care study, also known as the GPOC study. Between July and December of 2019, PATH, FMT, and ZPEM conducted a cross-sectional diagnostic accuracy study to evaluate the performance of the standard G6PD test and its ability to detect G6PD activity and measure hemoglobin. For the purposes of this presentation, I'll only be presenting the results related to G6PD activity and not hemoglobin, although if anyone's interested in those, they can be found in the full publication that I'll mention later. There were 2 study sites, Porto Velho in Rondônia, you can see on the map, and Manaus in Amazonas. And subjects enrolled in the study were individuals over the age of 2 who were recruited either from clinics as well as from an enriched sample of individuals with known G6PD status established through previous epidemiological surveys. For this study, the reference test for G6PD was a quantitative spectrophotometer at both sites, the point scientific assay, which was normalized by hemoglobin from a complete blood count automated hematology analyzer in Manaus and HemaQ in Porto Velho. At each site, there were 7 trained clinical research staff who were responsible for operating the SD Biosensor test. All operators underwent a robust study training that included multiple training sessions on clinical procedures, informed consent, as well as how to operate the test that included a presentation on G6PD deficiency diagnostic platforms, study procedures, demonstration and hands-on practice with the test using control reagents and discard blood, and as well as a competency assessment. Throughout the study, job aids were also utilized to support test operators in recall of key elements of test procedure. Next slide. So this slide shows an overview of the study workflow. Following consent and enrollment, a capillary finger stick specimen was collected from participants and was used to run the standard G6PD test and the HemaQ 201 at the point of care. A thick blood slide was also prepared for malaria microscopy. And then venous blood was collected in EDTA and transferred to the laboratory at each site where additional G6PD and hemoglobin tests were run. And as you can see here, those included repeats of the HemoQ-201 and standard G6PD test measurements on venous specimens. The quantitative point scientific reference assay was run. In Manaus, a complete blood count was performed using an automated hematology analyzer. And at both sites, the fluorescence spot test was also conducted as a commonly used standard of care test for G6PD deficiency. And importantly, all laboratory test operators were blinded to the results of the point of care tests. Next slide. Next slide. So getting into the results here, we're showing the demographics of the study population. In total, 1,736 individuals were included in the analysis, 924 from Manaus and 812 from Portobello. Participants ranged in age from 2 up to 92 years, and 55% of the study population was female. In total, there were 251 malaria cases in the study, the majority of which were from Portobello. And of those, 199 were P-Vivax cases. Next slide. During the study, the standard G6PD test was run over a broad range of operating temperatures ranging from 17.7 degrees Celsius to 43.7 degrees Celsius. And this is representative of true operating conditions for the device in a real-world malaria endemic setting. Next slide. These figures show the distribution of G6PD activity of the study population in percent activity as measured by the reference assay by sex. The frequency is plotted against the normalized G6PD activity values in 10% increments. Reference values here are expressed as the percentage of each site's adjusted male median, which was determined from 36 randomly selected males with G6PD activity within the reference range for the reference assay in all males established at each laboratory. These were subsequently excluded from the performance analysis. Quantitative thresholds for G6PD activity categories at 30%, 70%, and 80% were then calculated and used to assess performance between the standard test and the reference test. And you can see these activity limits shown on the graphs in the dotted lines. And as you can see, there's an expected bimodal distribution among the male participants as compared to a continuous distribution for females, which is consistent with the epidemiology of G6PD deficiency. Next slide. So to understand the ability of the standard test to discriminate between individuals with deficient, intermediate, and normal status, receiver operating curves were generated. Here we're showing the ROC curves and summary characteristics for both capillary and venous specimens for deficient males and females, as well as for intermediate females based on the 70% threshold. And the test was able to diagnose G6PD deficient individuals with a high area under the curve of 1 across both specimen types. At the intermediate threshold, the test also showed a high AUC at 1 for venous specimens and 0.98 for capillary specimens. Next slide. Linear regression of the standard G6PD test G6PD activity measurements as compared to the normalized reference assay results showed an R-squared correlation value of 0.62 for venous specimens and 0.56 for capillary specimens. And as you can see from these figures, the largest source of imprecision is observed in the high G6PD ranges, which lie within the normal range of G6PD activity. Next slide. In terms of sensitivity and specificity, these values were calculated for the test based on the manufacturer's threshold compared with the normalized reference assay values by specimen type. For G6PD deficient individuals, the test showed very good sensitivity at 100% for both venous and capillary specimens. For intermediate females, at the 70% cutoff, the test sensitivity was 96.9% on venous specimens and 94.3% on capillary specimens. On capillary samples, the standard test did misclassify 2 intermediate females as normals, which led to that drop in sensitivity. However, both of these participants did have G6PD activity levels of greater than 65%, which is within the variability of the reference assay. Next slide. So the agreement between the result classification of the standard test and the reference test was also calculated, shown here in 3x3 tables. Overall, the standard test showed good agreement with the reference test results at 96.7% for venous specimens and 94.1% for capillary specimens. And here you can, on the capillary table, you can see those two misclassifications of intermediate women that led to the drop in sensitivity. It's also worth noting that for one of these two female participants, the test was run on capillary samples at an operating temperature greater than the 40 degrees that's recommended by the manufacturer. But these cases are significant because there is an associated risk that the patient and their provider might be unaware of the patient's G6PD status, and as a result, could be provided contraindicated treatment, which might result in a clinically relevant adverse outcome. And it's important to monitor these types of misclassifications as part of future studies evaluating test performance or operational implementation in malaria endemic settings. Next slide. So just another way of looking at this data, these dot plots show the standard G6PD measurement on capillary samples on the x-axis mapped against the reference values on the y-axis for males. And if you click again, yeah, so the standard test overestimated the number of G6PD deficients, which you can see by this yellow box. So at the 30% threshold, among the 715 males classified as normal by the reference test, only 13 were misclassified by the standard test. If you could click again. The standard test did a very good job in discriminating between deficient and normal at the 30% threshold, as you can see by this green box, and all deficient males were correctly identified. Next slide. So now just showing the same figure for females on capillary samples. If you could click twice. So here you can see that the standard test overestimated the number of G6PD deficients and intermediates as shown by the yellow and orange boxes. And then click again. And also did a good job in discriminating between deficient, intermediate, and normal at the 30% and 70% thresholds, with only those two female intermediates by the reference test being misclassified as normal by the standard. Next slide. And lastly, we also did a bit of a sub-analysis to explore the relationship between malaria infection and the performance of the standard test. Here we show the correlations by linear regression of the standard test G6PD values compared to the reference assay for malaria positive subjects and negative subjects on both venous and capillary specimens. Among malaria positive participants, the R-squared value was 0.57 for capillary specimens and 0.54 for venous. And for malaria negative participants, the R-squared was 0.53 for capillary and 0.6. Slide. In terms of sensitivity and specificity by malaria status, the standard G6PD test also showed good performance for both malaria positive and negatives. And all differences observed here between groups were within the 95% confidence intervals. Next slide. Finally, for the 199 participants in the study who had P5X malaria, we also wanted to explore how the results of the standard G6PD test might have influenced their treatment for malaria. So of the participants who tested positive for Vivax malaria, there were 3 intermediate females, 1 deficient female, and 7 deficient males. All of these participants were correctly classified by the standard G6PD test on both capillary and venous specimens. And as you can see from this table, with females in the green and males in the brown, results from the standard G6PD test on capillary samples taken at the point of care showed that several additional Vivax patients with normal G6PD status were classified as either deficient or intermediate. 2 were misclassified as deficient, 1 male and 1 female, and 12 females were misclassified as intermediate. These misclassifications would likely have impacted the administration of 8 iminoquinolines to these 14 individuals until confirmatory testing could be conducted. But it's important to sort of mention this risk in the context of considering, the overall benefits of increased safe availability of radical cure treatment options to people who otherwise might not have had access due to their unknown G6PD status. Next slide. Next slide. So in summary, the key findings of this study are that the standard G6PD test shows good diagnostic capacity to differentiate between G6PD deficient, intermediate, and normal cases. All G6PD deficient cases were correctly identified by the standard test. All females with G6PD activity less than 65% were classified as intermediate or deficient by the standard test. And malaria infection does not appear to affect the standard G6PD test performance. Next slide. So we did just have a recent study published with these findings, all these figures, and some additional. So for further information and more details on the results, we've included a reference here to the publication that came out last month. Next slide. And lastly, I just want to close with a big thank you to our many partners and collaborators, a few of whom are listed here, and especially to the study participants and research teams at FMT and CIPEM in Manaus and Portobello, without whom the study would not have been possible. So thank you very much.
Many thanks, Stephanie. Your presentation was really interesting, and I'm sure that there will be a lot of comments and questions for you during our Q&A session. So I think we can move on to Diego right now. So Diego, many thanks for joining us and for presenting these findings today. Diego is a study coordinator at the Tropical Medicine Foundation in Manaus, Brazil, that works primarily with clinical and implementation research on G6PD deficiency. He also works on clinical research on anti-malaria drugs, snakebite envenomy, and SARS-CoV-2 infection. Diego is a PhD candidate and holds a master's degree in tropical and infectious disease from the Amazonas State University in the Western Brazilian Amazon. So Diego, the floor is yours.
Thank you, Roberta. Can you all hear me well?
Yes.
Okay. So I'd like to thank you all for your presence today, and also thank the diagnostics program for the invitation. Some of you may know me from previous presentations. I believe I presented some data from the CareStart, if I'm not wrong, last year. And today we will discuss now the usage of the quantitative test in the field. Stephanie presented the data from the clinical lab, and now we'll see some results we had from the implementation of the test here in 2 municipalities in Brazil. Next slide, please. So the protocol implemented 2 tests. At first, we planned to implement the CareStart, and then we decided to change the standard G6PD test because of its availability and increase the diagnostic performance. So the CareStart was implemented only in Rio Preto da Eva, which is a small city close to Manaus, between February and January, while the quantitative test was also implemented in Rio Preto da Eva and Macio Lima along the year of 2020. Next slide, please. So just to give you a quick introduction, we recently published the results from the CareStart implementation and it was neglected. You can see the QR code that you can access. And the main results we have was that we trained those professionals, we assessed their proficiency before implementation. We saw that the training was enough for them to perform the test and provide suitable treatment. And you can see that we had a subset of samples to confirm their diagnosis, and we saw that the test had 100% sensitivity and a 68.1% specificity, which could be quite low and could affect the malaria transmission profile by not providing the correct treatment for those patients. You can see that the treatment, the 4-hour treatment, was enough for the health workers to perform the test. And we also had a qualitative part in our study where we could access the perceptions both from professionals and from patients. And among all the data that we got, we could see that they complained a lot about the subjectivity over the test color interpretation. They also complained over the fact that the test should have been kept in the laboratory. For those of you who work with the CareStart, you know that there is a single buffer vial for the kit. And they also, we had a good acceptability of the malaria card. I will discuss it later. That's a small card that we use to put the results from the patients in this card so that they could bring if they had a medical appointment or a new malaria episode. Next, please. Here we have both study locations. You can see that they have different transmission profiles. Mansulima has more a urban malaria transmission profile compared to Rio Preto da Eva, which has more a rural transmission profile. So most of the units in Rio Preto da Eva were rural, while in Mansulima we had more urban units where the test was implemented. And for both municipalities, we also include the local hospital as a reference for possible cases of hemolysis. Next, please. So the study itself, we can divide it in 3 stages. The first one is the pre-implementation, which we train the healthcare professionals and assess the efficacy of the training. Later on, we had the implementation process, where we implemented the test and the healthcare professionals would perform the test at those different units before providing the Vivax malaria treatment to the patients. And during implementation, we assessed their perceptions both from the healthcare professionals and also from the patients. And after the implementation was concluded, we assessed the cost effectiveness of the implementation and also the recurrences. Next, please. So here we will talk about the training. We proposed at first a 4-hour total because of the work shift of the malaria workers that we have here in Brazil. We just decided to have a really concise and focused training for them to perform the test and to have enough information to interpret the test results, provide the suitable treatment, and also be aware and monitor possible signs and symptoms of hemolysis, and also what to do if they have a patient that presents some of these symptoms. So we had a theory section, which we showed a slide set. We also used a lot of job aids, which in this case was really helpful. That was something they really liked and used during implementation. We recorded a video training. We used case scenarios for discussions, and then we had a supervised practice in which around 5 participants had one supervisor for them to practice the test procedure. And then we ended the training with a theory and practice test in which we considered a passing grade of 90%. So they could only miss only 1 question in this practice and theory evaluation. Our target audience was malaria control workers that were workers that work in the malaria department within each municipality. And we also had some community health workers whose primary focus of work was not only the malaria itself, but they could also work with malaria, especially in rural urban. So we had most professionals as malaria control workers, but we also had some community health workers. And you can see that after only a single training section, at least 70% of the participants had the minimum passing grade, and the remaining needed some additional training for the theory or for the practice test. And I tell you that one of the parts that we had most difficult during the trainings was the part where they had to check the chip code that was inserted in the device and the chip code number that was provided in the kit. I will comment on that with you. And later on. And then other part was this part where they had to transfer the blood into the buffer and transfer the buffer into the device itself. They had a lot of trouble during that part. They did not hold the pipette as shown in the picture, but rather in the bulb of the pipette. So they usually had a lot of volume or too low volume to put in the buffer, and thus they had some errors during the procedure. Next, please. Here are some of the training materials we used. We tried to explore a lot of the training materials because we saw that during the qualitative interviews and focus groups, the participants really highlighted the use of these materials for a quick reference. Here on the left, you can see a poster that was pinned on the wall of each malaria unit. And on the right, you can see the malaria card that we provided for the patients, the green one for normal patients and the red one for deficient patients. So if they brought this card in a case of a new episode, the healthcare professional would not perform the test again, but rather just take the results from there and use in the national notification program. I'd like to remind that all of these materials, both the translated versions and the Portuguese versions, will be provided in the GOR website for you. Next, please. Here now talking about the implementation process, we can see the activity in our population. In total, we have 1,654 cases of Plasmodium vivax malaria, of which 66 were considered deficient, had less than 30% activity using the manufacturer's reference ranges. And I'd like, that is something I have discussed a lot with Dr. Gonzalo. I have over this fact that we usually see in the graph that we can see a curve between below the 30% and then another curve over the 60%, and usually between 30 and 60% between males, we see almost nothing. But here we saw some of the males also had this considered intermediate activities. If you press the slide once, please. Here we can see the data from the paper Stephanie just mentioned. This data was in the supplementary materials, where you can see the activity in their population, and we can also observe that there is now this tendency to see those intermediate results also in males. So that is something that we are planning to access further to see possible influences, but that is something that we can also discuss during the Q&A section. I know that there are other studies and it would be interesting to see what they have seen in those areas. Next, please. Here's the performance of the test in the field. From the total participants, we had samples from 140 patients. From these samples, we tried to genotype them and have the result confirmation through genotyping. And you can see that from the 44 deficient results we had on the test, only 21 were actually confirmed during genotyping, 20 as the African A-, which is the most prevalent here in the Amazon, when we also had one Mediterranean among these 44 females. So we can see that we had a sensitivity of 100%, both overall and for male and female sex. And we had a specificity of 80.7%. If we consider the whole population, we have a slightly increased specificity for males as expected, but a slightly decreased specificity for females. If you press the slide, please. And here we can compare the data from the care start that we implement.
I think we lost Diego. Diego, are you back?
Can you hear me?
Yeah, okay.
Okay, so next slide, please. Here is the same data from those 140 participants with two different visualizations. On the left, you can see the results divided by sex, and on the right, you can see the results divided by genotyping confirmation. And you can see that the test performed its job quite well in discriminating the real deficients among the other ones, especially on this graph on the right. You can see that all deficients are below the threshold for deficiency proposed by the manufacturer, while we had some of the participants that were misclassified in their normal column on the right graph, you can see. Next, please. I will talk now a little bit of the qualitative data. We have a lot of qualitative data, and I believe we would need a whole webinar just for this part. So I'll try to summarize for you some of the findings that we found here from our study. Next, please. So basically, our qualitative data was accessed using in-depth interviews and also focus groups, both pre-implementation and also during different parts of the implementation itself. But some things that we heard from the healthcare professionals was that they really liked the test because it was quick and easily read. And if we talk about Rio Preto da Eva, in which they also implemented the Care Start, they preferred the quantitative test because of the result. It's a numeric result and not a colored result to read, which they found quite difficult. Some even suggested they liked the test so much that some even suggested mass testing in the population, because this way they would have the activities from the whole population and they didn't have to perform the test every time a person had this malaria diagnosis. And they really highlighted the use of the patient cards. You can see in the top right corner of the slides, the red card and the green card, because these patients treated this almost as official documents. So they would really bring these cards to everywhere they would go, to the doctor and also to the health unit in case they had a new malaria episode. So we saw a good acceptability and a good usage of this card that could generate decreases in costs of testing and other things in the routine malaria diagnosis. Next, please. We found something quite interesting, because we saw that most of both participants and both patients and also the healthcare professionals associated hemolysis to dark urine. That is something that they really associated during the trainings. And it's usually the first thing that they see after they have this primaquine treatment. They knew that if they had presented some of the symptoms, including the dark urine, they had to look for medical assistance. But interestingly, they would not associate hemolysis to the treatment. They would associate hemolysis to other things, mostly the malaria itself, but also things they would consume during the malaria treatment, or other totally unrelated things that we will try to explain once we have the qualitative data analysis thoroughly done. Next, please. An additional analysis we plan for this implementation protocol is the cost effectiveness of the implementation. You can see on the right graph, just a preliminary graph that we have for this. And we will also try to assess the impact of the weekly primaquine in the weekly primaquine usage in the real field. I believe this is the first time the weekly primaquine was used in real settings in Brazil. So we will also have some really nice data to provide you later on this year. Next, please. Just to comment on some topics that we observed during implementation that were mentioned both by the healthcare professionals or the participants. One of the challenges that we have was the timeline for the Q&A. SD Biosensor did not, although they provided the controls, they did not provide one specific time frame to perform this test. So we have this, that is something that needs to be discussed within each system that will implement test, or either statewide or local or regional wide. That is something that needs to be decided first. We also had a wide range for the normal controls, but I believe it's quite high, 6 to 17. It can take a lot of things in between these, especially compared to the deficient control, which is only from 0 to 3. One of the things that were most complained about was the number of the easy tubes provided. They only provide the correct amount of tubes. So if you miss one of the points where you should mix the blood with the buffer and then transfer the buffer to the device itself, if you miss one of those parts, you will have insufficient amount of easy tubes to complete the whole kit. So that is something that standard biosensor might take into it, maybe add additional easy tubes, which I believe are cheaper than a strip to analyze that is missed to analyze with the G6PD. And something that is also necessary, and we observed here, is that it is necessary to train doctors and nurses, because the healthcare professionals would refer them to some of the units, but there they would sometimes misclassify that patient as a different clinical picture than the hemolytic anemia. So this is something quite interesting to consider in other implementation studies. There was something that we tried to do during the last year, but the COVID pandemic would not allow that. And one of the best opportunities we have was that all of this data that was generated here in SafePrim was used for the tafenoquine rollout study, which is known as TRUST, that just started in Manaus and Porto Velho. So SafePrim can be considered as a pilot study for the bigger one, which is the tafenoquine study. Next, please. This study was performed in an agreement between the Ministry of Health, the municipal and state secretariats of health. We would not have this study conducted if it was not because of this agreement, because we needed all the assistance they have, and it was a two-way collaboration. We collaborated with the trainings, materials, and they performed the tests in the field, and the things promoted in the protocol. I'd also like to thank the MMV and PATH and BMGF for their help during the discussions, during the implementation, and of course the participation of the patients and also the healthcare professionals, that without them this study could not be conducted. Next, please. So this is what I mostly wanted to mention you. We had a lot of data that will be coming up shortly in several manuscripts that we plan to publish within this year. And yeah, I believe that's the most important thing that I had to show you. Thank you.
Thanks, Diego, for your presentation. I think we can start the Q&A session, and I can see that there is a discussion going on in the chat already, but I wonder if does anyone has questions to Diego or Stephanie? Nope.
So, hi everybody. Diego, compliments on a great presentation, and it's really exciting work, and Trust has really benefited greatly from the work that SafePrim has done. Looking at the G6PD intermediate male subjects, I was just curious. As you know, we are planning to use G6PD cards for male subjects. So it'd be good to hear from you and from Marcus. What do we do for G6PD intermediate males? Are they going to be retested, or how would you suggest going forward with them?
I don't know if Marcus wants to comment on that, but for the SafePrim, the intermediate females were tested only once, and they received that as the diagnosis. But for the TRUST study, the tafenoquine rollout study, I believe that the females will be tested every time they have a new malaria episode. Just the males that will have the card that they can bring in a different time, and that the healthcare professional can use that result. But for the females, they will be retested. That is something that was decided before the tafenoquine rollout study. That would be tried to get the activity that the female participant would have at that time or at that episode.
That's right. And for male intermediate?
For male intermediates, we consider them in our scenario as normal participants, And deficient were considered only if they had below 4.0. That's the cutoff point for the provided by the manufacturer.
Thanks.
Thanks, Diego. Any other questions?
Could I just ask Diego, please, the percentage of males that you found had intermediate level?
I don't have that number here in my head, and I did not put that in the presentation as well, but that is something I will try to have and maybe try to update everyone on that in a future email, but I don't have it in my head now, the exact number of intermediate males.
Okay, thanks.
I saw we had some question also from Soy King. He asked about the quality control of the device when we performed it. That is something, as I mentioned during the presentation, that it needs to be discussed with each department and that will perform the test, but we try to use every 1 to 4 weeks. That would really depend on the supervision chronogram proposed by municipalities. They had these supervision visits, and we try to nest the quality control for the test within those supervisions so that it could be later, that we try to use the routine they already had for supervising other things within the malaria laboratory.
Thanks, Diego. Any other questions?
Hi, Roberta, I have one. Hi, Diego, Diana from the Ministry of Health. Thanks for your presentation, and Stephanie also. I'd like to ask about the weekly Primaquine treatment. Was it okay for the patients? If they have any concern about this new treatment? And this treatment, they have supervision of health workers at the municipality, or the Primaquine was just given to the patients?
In every training, we try to pass to all the healthcare professionals that this treatment should be supervised to monitor possible signs of effects. Of course, that would really depend on the personnel availability of each municipal department. But we observed that when they had these deficient cases, which were not a lot, they were more careful, so they would provide more assistance than the standard of care. So we trained them to supervise the treatment, but that lastly would only depend on the personnel availability for them. We will show that in the qualitative data in the manuscripts. We had some discrepant how can I say that? We observed different things between the participants. Some really liked the treatment because especially the ones that were previously hospitalized, they really liked because using the weekly regimen, they would feel nothing. But other ones would complain because it's too long of a treatment to have within 8 weeks. So that is something that we'll try to explore more in the manuscripts that will be created in the next weeks.
Okay, thanks. We hope the Primaquine study give us more information about the perceptions of health workers and patients, but we are really glad to start.
Thank you, Liana and Diego. I think there's a question to Stephanie in the chat. Could you share more about the details, frequency, cold chain, maximum travel time regarding transporting blood sample from point of care to clinical lab? How to account for the variability that may be caused by the transportation? Can you get that, Steph, please?
Yes, hi. That's a good question. So for this study, for the clinical validation study setting, it was done in a quite controlled manner. Following collection, specimens were brought back to both laboratories in cooler boxes at between 2 to 8 Celsius, and immediately placed into a refrigerator where they were tested with the reference assay within 72 hours.
Okay, thanks, Steph. There's another question for you. You mentioned malaria infection does not appear to affect the standard G6PD test performance. Do you envision other studies to get additional data related to this? Do you think it is necessary?
Well, that's a good question. I'm actually not sure if maybe Diego or Gonzalo have thoughts on this.
I'll hand over to Diego, and then Marcus. I saw he's on the line. He might be able to talk about studies about this better. But one thing I do want to clarify, when we say it does not appear to affect the standard G6PD test performance, is in comparison to the reference assay. So it might be affecting both equally. And then we do know that there's a lot of literature around if there recent hemolytic events affecting the G6PD activity in an individual. So it doesn't appear to affect the data, the performance of the test in comparison to the reference assay, meaning that both can be are affected equally in the same way if they are. That's the only thing I can add to that. And Marcus has just put a note about some recent publications from Rick Price and I think Ben Lay's group.
Thanks, Gonzalo. Unless anyone has any other question, I think we can get back to Amanda to share a few. Yeah, sure, go ahead.
I had a quick comment just to highlight. First, thank you, Steph and Diego, for the great presentations. The observation that, and some of the questions were related to this, the observation that Diego made is that there does seem to be a cost in performance of the test as we move from clinical studies to operational settings, and it results in a drop in specificity, which means that more people that should have access to daily primaquine or tafenoquine are not getting access. And I think it's important to understand that. I think the exciting thing is that the SAFEPRIM study was so detailed that there might be an opportunity to try to understand what the causes are behind that, but I think it'll be a little bit difficult to do that. But I think it's important to highlight that because we know that in Southeast Asia, people are seeing similar things. And the two studies combined showed that in the same settings, we observe the same thing.
Gonzalo, do you have an explanation or a hypothesis around that?
We all have lots of explanations. I don't know that any of them are substantiated with any evidence. So I think, Diego and Marcus's team have the most data to be able to explore that. And so maybe we should just wait until they've done their analysis. Sorry, that wasn't very helpful, Penny.
No, it wasn't. It really wasn't. But well, let's wait for Diego and Marcus to come back with something.
Okay, so any final questions, reactions about the presentations? Great, so I think I want to thank you all for joining us, for your questions, and also Diego and Stephanie for your presentations. And I will give the floor back to Amanda because she has some information to share with you before we wrap up. Thank you, everyone.
Thanks so much, Roberta. Yeah, just want to echo thank you again, everyone, for joining the webinar today. And thank you, Steph and Diego and Roberta. If you have any questions or suggestions, please feel free to reach out to gorcop@path.org or visit our website. And finally, I just wanted to note that the GORCOP is also hosting another webinar at the end of the month on a new tool for assessing the cost effectiveness of radical cure options. So please feel free to visit our website for additional information as well as the link to register. So thank you again and have a good rest of your day or evening. Thanks.